Ben-Tal, who will take us to the computational part. Ben-Tal has been involved in the development of all kinds of tools, now very popular in the scientific community. And today's talk will be on structure-based designs of drugs targeting the Influenza M2 channel.
So first I'd like to thank all who managed to establish this important center. And then also I'm honored and proud to be invited to give a talk in this conference that remarks the initiation of this important center. I will present our work on the discovery of preliminary effort to discover. Maybe this one, yeah, I should stand here. So, preliminary effort to the discovery of anti-influenza drugs. And the work was done predominantly by a very talented PhD student in maybe it doesn't work? Oh, yeah, it does. In Bar Fish, a PhD student in my lab, and others have helped, including experimentalists who tested the predictions. So influenza is one of the main diseases of the respiratory tract in humans. It causes hundreds of thousands of deaths annually. It also So in total, something like several hundred of thousands of deaths worldwide per season. And it's also the cause of major human pandemics throughout the world, including maybe the best known is the Spanish flu at the end of the First World War, where there's debate whether 50 or 100 millions of people eventually died, and that amounts to between 3 to 5% of the population at the time. So there is definitely a risk, and therefore definitely a need for a drug. It's caused by the influenza virus, which is the envelope of which has hemagglutinin and neuraminidase mainly, and it's and also M2, which is a proton channel, and that's going to be our drug target. The influenza is subtyped based on the hemagglutinin and neuraminidase, and this is just a short slide to introduce the life cycle of the virus. So this is the virus, and that would be the host cell, and the event starts with the drug binding to a sialic acid receptor, and that's the hemagglutinin is involved in that. Eventually, there is fusion of the membrane of the virus and endosome, and injection of the genetic material ultimately to the nucleus, and then viral reproduction of like a multiplication of the genetic material and reproduction of the virus, and then budding out of the cell with multiple copies, where neuraminidase is used to cleave the hemagglutinin from the sialic acid, and that's also an important part. The M2 channel is important for acidification of the virus in this stage, as well as in prevention of premature conformational change in the hemagglutinin at this stage. So the various approaches to prevent or to protect ourselves against influenza include inhibition of the neuraminidase and the zinavir that was mentioned earlier is one of the drugs here, but it's not very efficient, and it also involves you have to inhale it, which makes it very, you can't give it to a large population. It's not very easy to administer. The other alternative is to target the M2 channel, and the M2 is considered a good target because in contrast to many other components of the virus, it's supposed to be highly conserved, so it alternates less, and maybe in this respect a better target. However, for now, the only two drugs that are known are amantadine and rimantadine, but they are ineffective already because most of the virus developed immunity to the resistance to these. So here is what I have to say about the M2 channel that we target. So it's a short protein of something like 90 amino acid. It includes one transmembrane helix that tetramerize to form a proton channel. It is activated by low pH. It's a homotetramer, as I said, and it's critical for influenza infectivity, and therefore it's a good drug target. These are the known drugs, amantadine and rimantadine. However, because the virus developed resistance with a mutation, I'll soon touch upon this mutation. Oh, here it is. So there are several escape mutants, but the most dominant one is S31-N mutant, which dominates the viral population. So therefore, the CDC now recommends against usage of these drugs because they are no longer effective against this mutant. So we took an approach that is based on virtual screening, and in fact, it's based on a computational suit by Schrödinger that the center now has. So exactly what we did, we did it years ago, but exactly what we did is possible now to do for the entire Tel Aviv community. So, and that's the general approach. So docking tools, the problem with docking tools is that they don't always find the correct solution at the top scoring list. So one has to screen and look for it, and for that, one has to rely on chemical intuition. Had the scoring function been better, that wouldn't be necessary, but then it wouldn't be a challenge. So the challenge in this project is really that. And that's the big advantage where I benefited from Inbar, who has great chemical intuition. How do you evaluate the performance of this docking? So it depends on one case, between cases, but here it was easy relatively because we had a complex of the structure of the channel in complex with amantadine and rimantadine, so we could compare, and also there were known other derivatives that were known to bind or not to bind, so we could calibrate the result. So in general, you want to reproduce co-crystal structures with less than 2 angstroms. You want to reproduce interaction with important binding site residues. You want to rank active compounds better than inactive ones, and ideally predict the activity trend. So here is what we did for this project. So Inbar started with a focused library of something like 3,000 compounds that were, at least in size and in basic chemical properties, they should have fit to bind the channel, and she screened this library for docking and including docking of the amantadine and rimantadine and known inhibitors and non-inhibitors to calibrate the model. So she used the crystal structure in the open conformation of the channel, also a solid state NMR structure in closed conformation. And these were the only structures available at the time. That was something like 5 or more years ago when she started the project. And then she docked and looked at the results. And these are her top 3 poses for docking of Amantadine. But that was not the original result. She had to play with it, to play with the scoring function and the various aspects of the procedure until she developed a procedure that made it possible to reach this result. And that's the best score. So it's like it has a property of free energy, the scoring function. So the more negative, the better. So that's the best score. And here is a comparison of this best score versus the known co-crystal. And you see that it's very, very similar. Then she did the same with rimantadine, and both molecules bind such that the charge amine point downwards towards the so-called his gate for 3 histidines, 4 histidines that are there, 1 from each subunit. And again, that's the best score. And it also superimposed well with the co-crystal. So now that she managed to reproduce as much as possible the known co-crystals, she went ahead with some known binders, and that's docking to wild type in green, and you see that all of them dock well. She also did homology modeling of the mutant, of the escape mutant, and the results are in yellow. And you see that very encouragingly, they dock much less well to the mutant. Rimantadine is especially nice in that it docks with positive energy. So that's another feature that her model reproduced, to some extent at least. Now, she went ahead, and that's possible when structure is known or predicted. She went ahead with developing some chemical intuition about the results. So what it is that makes the difference between the wild type and the mutant. So this is the crystal structure with binding of the wild type, and that's the amantadine molecule sitting here, and you see that it nicely fits in a binding pocket. Now with the mutant, the binding pocket gets smaller, and also it becomes less hydrophobic, explaining perhaps why the mutant binds less well the amantadine. So with all of this in mind, she felt comfortable in doing the screening. And as I said, she started with 3,000 molecules with focused properties, and having developed this 3-dimensional pharmacophore model, she just screened all of them against the same model. I repeat that the point is that she uses the exact same protocol that made her reproduce the data for the known to now look for putative unknown binders. And again, she used the crystal structure in open conformation, as well as the solid state NMR structure in the closed conformation. And with that, she came out with 74 binders that looked good. Now, coming out with the 74 is very challenging. So for that, she looked at, from the results, she clustered them into thousands of different clusters with chemical similarity, and she looked at the representative complex, or docking complex of each of the clusters. So I don't know how many of you looked at complexes, but it's very, very tiring to look at thousands such complexes. I look at few and my patient, and then I run out of patience. it's very, very difficult to look and realize what is chemically right and not. So I appreciate this part of the work a lot. And that's how she compiled this list of 74 compounds. She then went ahead and docked those 74 versus the model of the mutant and came up with a list of 14 compounds. And later, she also, in collaboration with experimentalists, they also looked at SAR homologs and synthesized some, and all of this was tested. But this is a description of how the experimental test went by. So they used two influenza strain. One is called PR8, and that's like the representative of the pathological influenza. There is also a BLE, which does not have the exact same Amantadine, the same M2 channel, so that should be insensitive to all the drugs. And that's the sequence of M2 for the two variants, for the PR8 and the BLE. The specific strain includes the mutation here. You see this N, which replaces the S. And that's what the results look like. So there is the assay is called the Munana assay. I hope I pronounce it well. Anyway, the point of this molecule, of this assay, is that it counts that the Munana thing is cleaved by the neuraminidase. So that's a way to quantify the number of neuraminidase in the system, and that scales with the number of viruses. So the more viruses there are, the stronger activity there is. And based on that, it's possible to measure blockage of the channel. So these are the 14 compounds that she used. The 4 ones that are good, I'm not allowed to show. So, and here are results of those. So there is series, we mark them as series because we use SAR, so that would be series 1, 2, 3, and 4. So this is series 1. So this is the original compound, and that's the readout from the activity as a function of concentration. So this is in red. That's the BLE. So that should not feel the compound, and it doesn't essentially. That's the PR8 that should sense the compound and should be sensitive to the compound, and it does, as you can see. And these are analogs, so analog number 2 and so forth. And you see that they are all active. They all inhibit. Now here is the two particularly interesting analogs, analog number 2 and 4. They are very active. And here is a comparison to Amantadine as control. And you see that Amantadine essentially doesn't sense, doesn't bind. And that's because of the mutation that makes the channel insensitive to Amantadine. So you see, and like the take-home message from this slide is that these two analogs are better than Amantadine. So they are able to bind also to, theoretically, to inhibit virus with the mutation that dominates the population now. This is series 2. And again, so this is the original compound that inhibits a little bit, and analogs that inhibits more. And in series 2, there was also effort to do synthesis. So these two are synthetic compounds that inhibit better. And again, there is Amantadine as a reference here, so you see that all of them are better. The synthesis had improved activity even further. Here you see again the synthetic molecule that did better. We don't have enough data to estimate by how much, but you see that they do better. This is series 3, and again the original compound and analogs, and you see consistent improvement with the analogs. I think I can skip this slide. And again, series 4, you get the idea basically. This is a control experiment to show selectivity, to show that the compounds themselves, I mean, theoretically, they could directly bind and inhibit neuraminidase. This slide shows that they don't. So, and this curve here for DDAN shows active inhibition of neuraminidase, because that's a known inhibitor of neuraminidase. None of the compounds show similar inhibition, so you see that they don't interact directly with neuraminidase, so presumably they interact with, they block the channel and therefore inhibit the virus. And that's another control to show that there is no effect on the hemagglutinin, which is also outside the virus, so theoretically it could interact with the virus. So here is, so it's an agglutination assay. So this is an antibody that inhibit hemagglutinin, and you see the agglutination, the prevention of agglutination as the red dots here in the cells. In the compounds, you don't see this effect, so they presumably don't interact with hemagglutinin. And these are our plans for continuation here. So we want to examine our promised hits using more assays, in particular assays that examine specifically the activity of the channel, the proton conductance. So we want to see that directly. We also want to optimize the lead compound using structure-based design, which we started, but we want to do more. There are now more structures, and we are trying to look at those and improve on our compounds. We also feel that we need to do co-crystals of the compounds of our hits with the channel, and we feel actually that this is very important because our preliminary effort with the compounds and the structures, we see a lot of inconsistencies there in trying to give molecular interpretation of the results. So we feel that we need, so maybe the binding mode of our docking model is less perfect than we would have wanted. So we need another structural anchor to proceed the project. And then we need to do preclinical ADME studies, and hopefully with the help of the center. And acknowledgement, so you already saw the people involved. From my lab, it's in Bar Fish, who did most of the work. Ori Khalid was also involved. From the experimental side, it was mostly in Philadelphia, and those are the people involved. I'd be happy to answer questions.
Do you hope that we can help with the medicinal chemistries?
So I have two questions actually. So the first one is whether your compounds are active also against the wild type.
Yeah, it's missing. We should have measured it, but it's still not done. Yeah.
And the second question is whether you have tried to see how easy or how difficult it is to raise resistant antivirals against these compounds?
Yeah, it's another thing that we should do. One thing that I should have mentioned, these are very different scaffolds from the amantadine and the rimantadine. So because they are different, then maybe there's hope that there will be, whatever the reaction of the virus will be, it's going to be different to the reaction that it has against rimantadine and amantadine. But I,
You could have another
yeah. Right. Again, I should stress that in principle, this channel is conserved, but nevertheless there were resistant mutations. So presumably there will be also for our molecules, and this is something that we should estimate. Yeah.
Thank you very much. Thank you all again for taking part in this first symposium, for many more to come in years to come. I took the advice of Ralph to be approachable and to run after different academic projects. So this is the first of many attempts to get people. I see here people in the Tel Aviv community which are not yet involved in drug discovery and development efforts. So we are, it's wrong to say in Israel, but like a crusader trying to get more and more members of the community take part of this activity, and hopefully in the next year, and we'll show some of the results of the first year effort. Thank you again, and see you next year. And also, and I would like to advertise another meeting that will be a scientific one on new horizons in drug discovery for rare disease. It will take place on September 8th in Bet Atfutzot lecture hall, and you are all invited to join this as well. Thank you.